Good day, everyone, and welcome to another screencast here in microbiology. I am your professor for today's session, Dr. Supachay Basit. And again, for today's session, we are going to discuss bacterial morphology. So, bacterial morphology depends, of course, when you say morphology, we are actually pertaining to the size and shape of the microorganism. Now, a typical bacteria would measure between 0.4 to 2 microns. Okay? If we're going to relatively compare the size of the bacterial cell on a logarithmic scale, so bacterial cell is almost the same size of the mitochondrion. Okay? But viruses are even much smaller, okay, than bacteria. Whereas the plant cell is between this scale, a frog egg is between this scale, and this is the ostrich egg, an adult female. So you can just imagine, okay, the size of bacteria relatively compared to other cells. Now, going back, the microscopic shape of the bacteria may be classified as cocci, bacilli, or spirochete. Okay, so this is how we classify the bacterial shape. Okay, so we have here the cocci. So we have the spherical shaped bacteria. So there are several variations of cocci according to their arrangement. So you would notice that some of them are arranged in pairs. We call them diplococci. An example is Streptococcus pneumoniae. Some of them are arranged in grape-like cluster, so Staphylococcus aureus. Some of them are actually arranged in chain, so we call it the Streptococcus, Streptococci. By the way, cocci are the plural form, coccus is the singular form. So Streptococcus pyogenes is an example of Streptococci. We also have the tetrads. They are arranged in 4. So Mycococcus tetragina is an example of tetrad. And then Sarcinae, they are arranged in a cubical packet of 8. So Sarcina ventriculi is an example. And then we have the rod-shaped bacteria. So some of them are arranged in chain. Some of them are flagellated and spore former. So an example of bacilli arranged in chain is Bacillus anthracis. And flagellated rod is Salmonella typhi. And then spore former Bacillus is a Clostridium botulinum. Bacillus is the singular form, bacilli is the plural form. Now, the spike-shaped bacteria, some of them are in curved shape. So, Vibrio cholerae, so Vibrios, comma shape or curved shape. Some of them are in spiral shape, spirilla, such as the Helicobacter pylori. Some of them are also spiral shape but has a corkscrew motility because of the presence of the eggshell filament or the endoflagellum. So an example is spirochetes such as the Treponema pallidum. Now going back to the shape, there are some shape that are said to be a fusiform, such as the fusiform bacteria, and palisading. When you say palisading, they are arranged in side-by-side arrangement. Now, a bizarre shape is this one, Haloarcula quadrata. So this is a rectangular shaped bacteria. And then we also have a star-shaped bacteria, Stella vacuolata. Okay. Now, as regards to the arrangement, I think we have already discussed some of the arrangement. Okay. And we also have here the one that I did discuss, the palisade bacteria. So Corynebacterium is an example of a palisade bacteria. And then we have the spore forming, I think I did mention in the previous slide, Streptomycetes. This is the mold-like filamentous bacteria. Okay, so these are some of the bacterial shapes and arrangement. So another variation that you can see here. So we have a transition between cocci and bacilli. We call it Coccobacillus. If the bacilli are arranged in pair, we call it Diplobacilli. If they are arranged in a chain-like manner, we call it Streptobacilli. So there are also some budding and appendage bacteria such as the hypha and the stalk. Okay, so the fusiform bacteria is the enlarged rod. Okay, so these are the bacterial morphology and arrangement. So moving forward, we have also the different stains that we can utilize for bacteria. The most common of which is the gram stain. Okay, the gram stain. So I will be including in this particular module the staining technique that you can actually utilize. So gram stain is one of the major stain that divides the bacteria is as to gram positive and gram negative. So we are actually using 4 stain. Crystal violet serves as the primary stain. Grams iodine serves as the mordant. And then the decolorizer is the acetone and alcohol, and safranin is the counter stain. So a gram-positive bacilli will have a deep purple color. A gram-negative bacilli would have a pink, okay, or red in color. Acid-fast stain, I think I did mention this to you. Acid-fast stain is the ability to resist decolorization by acid, alcohol, and there are 3 main methods of doing it. The heat method or the steam method is the Zille-Nielsen method. The Zille-Nielsen also utilizes carbonfuchsin as the primary stain. Now, the Quignon's method is not the heat or the heat method or the steam method. It is the cold method because here, instead of adding heat, we add detergents such as tergitol, and we prolong the contact with carbonfuchsin. So carbonfuchsin still here serve as the primary stain. And then the decolorizer is also an acid alcohol, but this time the counter stain is also a methylene blue. So an acid-fast bacilli will appear red, while non-acid fast would appear blue. So a more sensitive method is the auramine rhodamine stain. So these are examples of fluorochrome stain wherein you have to use a fluorescent microscope and the organism will glow in the dark. So this is an example of an acid-fast bacilli. So you can see here the red or pink stain. So it is actually against a blue background because the blue color there means that those that are non-acid fast had actually taken up the counter stain, which is the methylene blue. So this time it is an example of an acid fast stain that has been stained with a fluorochrome dye. So acridine orange is another example of stains. So it stains the nucleic acid bright orange whenever you are using ultraviolet light. So it will tell us whether the bacterial cell is still living or dead. So it is used to locate bacteria in blood cultures and other specimens wherein discerning bacteria might otherwise be difficult to see. If the organisms are actually dead, it will take up the stain and you'll be able to see an acridine orange, whereas living cell will not be able to take up the stain. So here is an example of an acridine orange positive bacteria. Calcofluor white is actually a whitening compound in textile industry. It is actually being used as compound that binds to chitin, which is actually one of the components of the fungal cell wall. So it appears like a bright apple green and blue-white fluorescence when observed under the ultraviolet light. And other stains that can be used are the following. Methylene blue, it can stain the metachromatic granules of Corynebacterium diphtheriae. So we call that the Babe's Ernst granules. Then the lactophenol cotton blue is used to stain medically important fungi grown in the Henrici slide culture technique. So it will stain the fungal cell wall blue. And then the India ink stain, the one that I've discussed in the previous lesson. So it is a negative stain that can visualize capsule. It is because the negative stain will stain the background instead of the actual capsule, wherein the actual capsule appears to be colorless. And then the endospore stain, so is the most common example of an endospore stain is the malachite green. So here's the methylene blue that stains the metachromatic granules of Corynebacterium diphtheriae. then lactophenol cotton blue stain, which stains the fungal cell wall, the India ink stain, which stains the background and makes the capsule colorless like halo surrounding the cell. Okay, so that's the end of our discussion when it comes to bacterial morphology. Please watch the animation together that I have included together with this module. That will be all. Wherever you are, God bless everyone. Stay safe.